Hi, everybody. Welcome back. We're going to be talking about our innate defense mechanisms now. We're going to look at the first line and then the second line of defense. So, first line of defense is your skin and mucous membranes, and then the second line of defense are the internal cell structures and chemicals released that help to protect our body before our immune system has to even come into play. So, the skin, epidermis and dermis, is part of that first line of defense. Included with the skin, we have microbes that are living on our skin. So these are part of our normal flora, and they actually keep pathogens from being able to survive. So think about if if you're living at your house and someone else wants to move in, you're going to fight them. And that's exactly what our normal flora does to pathogens. Pathogens try to take up real estate on our body, and the normal flora is going to outcompete. And there's more of those normal flora than there are the pathogens, so they're going to win. We also have chemicals released by our skin, our sweat glands, and our sebaceous glands release chemicals that are going to kill off pathogens or get rid of potential pathogens or keep pathogens from being able to replicate. We also have a mucous membrane. Mucous membranes is the layer of tissue that lines all of the body openings that are open to the external environment but are still moving inside. And mucous membranes produce mucin, which is a chemical that, when hydrated, forms that mucus, and mucus typically keeps bacteria from being able to replicate. So there are, again, we have millions of bacteria that live within the mucous membrane that are going to outcompete other pathogens, and any pathogens that do get within that mucous membrane are going to be suppressed from growth because they're not going to get the nutrients they need, oxygen, or they're going to be killed off by the other cells that are there. So barrier mechanisms are pretty successful, but if something gets through the barrier, that's when our second line of defense, as well as our adaptive immunity, is going to be activated. So these two get activated really at the same time. So let's look at our second line of defense. We have neutrophils. Neutrophils are the most common white blood cell that we have. Neutrophils function in phagocytosis, but they are not your professionals. So when they phagocytize something, they basically can only kill off one or two items, and then they themselves will die. Macrophages are your professional phagocytes. And so they're going to be able to hang around, and they get rid of everything in that environment. So neutrophils will arrive first at the site of an infection, but macrophages will stay longer and really clean everything up. Both neutrophils and macrophages function in phagocytosis. So they both produce what we call a phagosome, which is that vesicle that is produced via endocytosis. That phagosome then will merge with a lysosome and now it's called a phagolysosome. The lysosome releases digestive enzymes and breaks down whatever's in there. The phagosome can also merge with a peroxisome and release oxidative agents that will oxidize that pathogen. We have basophil and mast cells. These function similarly, but they're located in different areas. Basophils are found in the blood. They are the least common white blood cell. And then mast cells are found in our connective tissue and in our mucous membranes. They both produce chemicals called heparin and histamine. Histamine is your vasodilator and it increases capillary permeability. So it's going to cause or stimulate an inflammatory response. And then heparin is an anticoagulant so that fluids move faster and don't stop moving. They kind of become more slippery in a sense. And so fluids can move faster, easier to that site. So you're going to get a very large inflammatory response, which with the inflammatory response, there are, you always have chemicals that are released stimulate other white blood cells like your neutrophils, like your um, macrophages to come to that site as well. And then they release eicosanoids, which increase in the inflammatory response as well. We have natural killer cells. Natural killer cells are lymphocytes, but they are non-specific lymphocytes. So they're going to destroy a wide variety of different cells that are infected with some type of pathogen. So these could be either virus infected or bacteria infected cells, or they could be cells that are growing out of control. So they have some form of like a cancer where the cells um, self proteins are mutated, and so they're not growing properly. And so natural killer cells will attack all of those types of cells. So they're not specific, but they are what we call immune surveillance cells. So they move through our body continuously and they detect any cells that are considered unhealthy and then they kill them. And they do this by releasing cytotoxic chemicals. One of those chemicals is called perforin. Perforin is going to produce holes inside of the plasma membrane, and then granzymes can move through those holes, those perforations, and they induce apoptosis. We have eosinophils. Oh, I will mention quickly. If it's underlined in blue, then there is a link to a video that you can watch, okay? We have eosinophils. Eosinophils target parasites, specifically helminthic worms. So helminthic worms, that's like saying worm. Okay. Anyways, they target worms that live in your gut, and they release chemicals that break down the worm body. They also can undergo phagocytosis of specific worm antibody complexes. And then we have interferon. Interferon is a group of chemicals or antimicrobial chemicals that are produced by most of our body cells. And these chemicals are going to be released when our body cells get infected with a virus. So interferon, once it's released, binds to our neighboring cells and prevents them from becoming infected with the virus. It does not protect the cell that produced it. So if you produce the interferon, you're basically sending out a message to all the neighboring cells, don't open your door, there's a pathogen around. And so that cell's still going to die, but the other cells will live. The chemical also stimulates natural killer cells and macrophages to come to the site of the infection. And so this is something I made to try and help people understand. These are the different chemicals or cells that I have, and then these are body cells. So here we have a couple cells that are getting infected with a virus. The virus is going to start spreading or replicating inside using the cell's machinery. those cells are producing interferon. The interferon goes to all of our neighboring cells and it causes these cells basically to keep their doors shut so the virus cannot get in. What that does is these cells are now protected. These cells are not protected. So because of that, hold on. Okay, I thought there was two of these. If I click again, is there another one going to come over here? There we go. All right. So what happens next, natural killer cells and macrophages come to the site. Natural killer cells release those perforins and granzymes, which cause apoptosis of our body cells. And then the macrophages come along and eat those damaged cells. So now let's talk about the complement system. Complement proteins are a group of proteins that are produced by the liver. They are inactive in general in our blood plasma. There are millions of complement proteins floating through your blood all the time, but they're not activated, which is a good thing because otherwise they could have hurt our own cells, which would not be good. Okay. Each complement protein is named with a letter C to identify the complement and then a number after it, okay? There are two major ways the complement can get activated. Get this to move. You have a classical pathway or an alternative pathway. In both cases, you're going to eventually activate C3, complement protein 3. So in the classical pathway, an antigen and an antibody bind, and that activates C3. C3, when it becomes activated, is going to split into two molecules, C3A and C3B. In the alternative pathway, C3 is every once in a while just converted randomly, and if it binds to a pathogen directly, then it's going to become very active. So if you randomly produce hydrolyze C3 through C3A and C3B, normally they will break up down very rapidly. But if they are bound to a pathogen when this happens, that activates the alternative pathway. Okay. In both cases, we're going to have different, so we're going to have similar events occur. So in both cases, multiple responses of the complement system will happen. One of those is called opsonization. Opsonization occurs when complement proteins bind to a pathogen, and the binding allows your phagocytic cells to attach and endocytize them easier. So it's kind of like, I always think of opsonization like adding ketchup to fries. They're more desirable, and so phagocytic cells can eat them easier. They're going to cause inflammatory responses. So they activate mast cells, basophil cells, to release chemicals. And in doing so, they attract your neutrophils and macrophages. They're going to cause cytolysis. So the cell of the pathogen is going to lysis open. This is known as a MAC attack or membrane attack complex, when complement proteins basically poke holes into the plasma membrane. And that allows fluids to leave the cell, and so internal fluids of the cell to leave and materials from outside to go in. And then we have the elimination of potential pathogens or toxins. When antibodies and antigens bind to the pathogen, they can then be eliminated going through the spleen, so into the spleen, and you have those phagocytic cells that will just get rid of those materials. We have another response, just inflammation, which I talked about in the complement system, because complement can activate inflammation, but inflammation in a nutshell doesn't have to be activated by complement. Basophils and mast cells are what typically activate the inflammatory response. And so what happens is the cells release chemicals, histamine and heparin, which increase the amount of fluid flowing in that area, which is what causes that swelling, and make the capillaries more permeable. So more fluid will move to that area. But those chemicals also stimulate your macrophages and other white blood cells to come to the site of the infection, neutrophils, macrophages, to get rid of the potential pathogens. So it basically keeps the pathogens in one area and allows your white blood cells to kill off the pathogen. And one of the last innate responses we'll talk about then is fever. Fever is an elevation above normal body temperature. So normal body temperature is about 37 degrees Celsius. Fever is anything above that 37 degrees Celsius, typically about 1 degree Celsius above that normal. So if you're like 37.1 degrees Celsius, you would consider yourself having a fever. Fevers often are associated with other innate defenses. So if you have inflammation, you may also get a fever. That's part of the body's mechanism of trying to fight off the infection. So fever results from the release of pyrogens. Pyrogens are chemicals that stimulate your hypothalamus to increase body temperature. So fevers are really beneficial. Fevers inhibit bacterial and viral replication, but they activate interferon activity. Interferon is what protects your body cells from viral attack. It also increases normal tissue repair, normal adaptive immune responses. Anytime you warm up, then your body can work a little bit more effectively. So a fever is beneficial so long as it's not too high. And that's where this picture, I just thought it was adorable with germs on Motrin. They're pretty happy, but germs on a fever are not very happy. But if your fever gets too high, so typical fever above 39 degrees Celsius or 100 degrees Fahrenheit, or if it's so above 100 degrees Fahrenheit is considered a high fever, but it's not considered deadly. In children, if it's above 103, and for adults above 101, then you start worrying a little bit about the fever because that's a pretty high fever. If you get a fever that is above 106, that could lead to irreversible brain damage. And if it's above 109, that could lead to death of the individual. And so why does this happen? Because remember, all of our cells are composed of proteins. All of those complement proteins in our blood are proteins. Antibodies are proteins. Proteins function at specific temperatures, and they denature when the temperature gets too high. So denaturation of those proteins causes those cells not to be able to function anymore, which is what leads to the brain damage and potential death of the body. I'm going to stop here, and we'll get into the adaptive immune system in our next video. Bye.